Sorry about. Okay, let's get it started. So this is week number 7, halfway through, just before the spring break. So I wish you learned something over the last couple of weeks, and if you believe there's a better way to make your learning more effective, let me know so we can adapt the new methods. So as I said, I try to put what I on my mind to your mind. The best way is to let you to record you repeat the material in the lecture and to see how you really understand. I enjoyed to watch your homework video. And some of your video is too brief, just a couple minutes, 3 minutes at least. But it's okay. You get the idea. So this week we are going to discuss classification. Last week we discussed clustering. Both of those are the topics data science really like, and they did a good job in the machine learning field. So clustering last week we discussed was a unsupervised method. And this week, the classification is a supervised machine learning, like standard. So we will discuss a couple of these commonly used classification methods, including neural network, KNN network, and logistic regression I will mention, linear descriptive analysis, naive Bayesian, the approach, the framework. So in the middle of this general discussion of those general methods in classification applications, I will mention the one using neural network to do single cell type classification, one of the recent paper published. Then we will mention a couple of methods people use to evaluate the effectiveness of classification method. So you build a classifier algorithm and train it. Then you need to have a standard to see the performance of your pipeline, your classifier you build. So there's a method you can see, okay, this is a good one, this is a bad one, or this is a so-so. At the end, I will mention the cell type classification or identification method we built in the toolbox. So what is the classification? So this is a large group of methods that help you to perform this, conduct this process to predict a class of the given data point. As I said, it's very different from clustering. Clustering, you don't have to train the model. You can let the algorithm to cluster data points for you. Well, the classification, you need to prepare the training data first. In this case, you provide two classes of data points in labeled. Okay, you have labeled data points first, and then you train the model to learn there's separation, which indicated by this dashed line. Once you finish the training, the model has been built, and then when you have a new data point comes in, and then you will see, okay, this data point located here, and it belongs to the gray data point class group. If the new data point comes in and it's located here, and then it belongs to the red group. So that's why the classes are called targets or labels or categories. Labeled data has been valuable recently because you need good training data to train the neural network. Neural network is a class of these classification tools. Supervised learning, okay? So you need to take 2 steps. You need to train the model to let the algorithm to learn. So this is a learning step. And then you do the classification. You test your new data points. Okay, so the model or the trained model is called a classifier. It can be in many different forms. So that's why there are different categories of the methods. Another example, illustration of this machine learning framework of the classification. Label the data as a training set, and then you can, in the middle, you can do the feature extraction to see, okay, you don't need to use the full dimension of all the data variables, but you only pick those important to train your model. And also you put all the data into vector form. So whatever machine learning classifiers, this algorithm will take data input as vectors. As I said, we treat all the data points, data as vectors. So that's why from the very beginning, we vectorize our data points, either 2 numbers in 2D, 3 numbers, high numbers, you have high dimensional data. Even you want to put pictures into the machine learning tools, like to do the training or testing, you have to vectorize your image. Image is 2D. If you colored your multiple dimension, you have to find a way to do the vectorization before you put the image files into the machine learning pipeline. So this process is called vectorization. No matter nowadays, no matter what you are doing, you always have to do that. Other models of the data points, you also have to give the machine the vector, because that's this computer framework can take. In the future, maybe we have different kinds of forms of computers, not like a digital computer. Maybe you have analog computer, you have a quantum computer, may take different kinds of inputs, okay? So once you train the model, your supervised learning process is done. You put your trained model, the classifiers, and into operation. And here you can start to apply this classifier to the real data. So in the middle of these classifiers, you need to build a function. So this is a very general description. So this function will take your input, okay, give you the output as a label or classes, okay, or categories. You gave a picture of apple, it gave you the label. So apple is one of the categories of the output. You gave a tomato, then gave a picture of cow, and I gave a cow. So this is a trained model. The function will digest information provided from the input. So when we say we are doing the training, we basically provide a lot of inputs, a series of data points, to let the machine to change the function of f parameter, to make the f customize the function. Okay, the function framework depends on what type of the classification algorithm you use. there's always a function in the middle. This function could be a series neural networks, a lot of neurons combined. It could be a logistic function. It could be a Bayesian framework, and there's a function in there. It could be the LDA, okay, their function. So you train the function to change not the form, but change the parameter of the function to make it ready to make a prediction. So we gradually, from this illustration, and gradually introduce the function, the data, the label, so you start to see the mathematical expression. But the whole idea are the same as we illustrated cartoon figure like this, and the function or mathematical expression are the same. This is just more abstract. If you search Wikipedia classifiers, machine learning, you will have a long list of many different algorithms have been built over the years. As I said, classification is one of the best solved problems in machine learning. There are so many applications in machine learning field using classifiers to do the job. So give a picture, you will see, okay, this is a cat and a dog. So people always use those kind of examples as a machine learning examples. So that's why, because they are well solved. There's so many options in there. In computer session practice, we will use MATLAB. There are built-in classifiers, maybe a dozen of them, different categories. Within these categories, there are different subcategories, and you can choose from, and to train the same training data and see which model performs the best. So we will try that. But today we just go through some of the general ideas behind each categories of those most commonly used classifiers, okay, including logistic regression, Bayesian, perceptron, and which is the fundamental unit of the neural network. Next week, after the spring break, we will mention support vector machine. There's also decision trees, other methods as well. Okay, perceptron. It's a neuron in machine learning. Okay, people in the computer science field, several decades ago, they have a very early idea. We want to use this simulation to simulate a neuron to process the data, process information to make a decision. This is like borrow the idea, borrow the knowledge from the biology, neuroscience, especially. You take inputs from different axons here, and then aggregate information to give the neuron, and the neuron will reject the signals and as output, and pass on this information to next neuron. So we are looking at the structure right now in the middle to see the most basic unit of this neuron is simulated in the computer. Okay. I will say at the end, this will be mainly operated in the matrix multiplication. Okay. Let's see, we have an input data. Okay, our input is a vector. Okay, vectorization. As I said, everything has been vectorized. So you have input data. So this data. So let's see just one vector, and this is like 4D data. So I will have x1, x2. This can be higher, okay? But in this case, we just think, okay, this input data is this. To build a neuron or perceptron, we need to have another vector. So this vector is called a weight. So we have a weight here, another 4 numbers. So another vector. So this vector will be weight 1, weight 2, we will do so 2 vectors. So you have your x vector x, and you have your weight vector. We are going to do a dot product. A dot product will be so how can we do the dot product? We will put this is 1 by 4 and 4 by 1, right? So we will basically transpose this horizontally, make it, and then when you do this dot product, it becomes x1 times w1 plus x2 plus x3 x4. So this is a dot product, right? So the output of this dot product between 2 vectors is a vector, is a matrix, or is a single number. The dot product should give you a single number in this case. So as we expressed here, so those dot products give you aggregated information into a single number. A single number is this dot product between your x. Yeah, our case is 4 dimension, but if you make n and you have n, it's the same, okay? You just need to have a more parameter to change. So we have a single number here, and then this will pass on into a function, so it's called active function. Activation function. So this function basically just gave a transformation of this point, this single number. And to transform, there are different forms of the activation function. If you look at this, one of the active function can be something like this, okay? If the value of the input, if this product is small, it's a negative, then this output will be 0. If it's positive, it will give you the linear increase of the value and translate into y. Or there are some other forms, okay? You can use this S-shape function, sigmoid. There's a soft cutoff. Once you pass on this threshold, you suddenly become 1. Otherwise, it keeps close to 0. There are different ways to transform your single number to give you the output. So we just give you the output. Okay, so this is a very simple, just one neuron. We can take input and aggregate information, give output. Let's say I give you a small example to illustrate how we can use this. Okay. In this particular example, so I said there are 3 inputs. So this is the first point, data point, okay? Just for random number, basically I made it up. And then you can see, okay, if I set up my neuron into this state, set 1, you can put 4 numbers of weights into 1, -1, 0, 0. In this case, if we label the data, say, okay, the data should be, if I give the input of this, do the dot product, I should give -1. This is my labeled data. So if I know this first data point output should be -1, then the second should be -1, the third will be +1. So that's what I expected. I want to train my model. I want to set a parameter set to give me this kind of output. But if I use the set 1, I try these 3 numbers, and it will, this set 1 will give me positive, -1. So this is not what I want. So I have to retrain the model until I get some numbers like -0.7 as a weight 1, 1.4 as weight 2, 0 as weight 3, 0.7 as weight 4. When I set my neuron in this way, then I try these 3 training data. When I try the first column of data, okay, the first point, it will give me -1. The second will give me -1. The third will give me -1. Okay, so that's my expected. I train the model to make right prediction for my training data. The 3 training data gave me -1, -1, +1. Then I would see this data set 2 is well trained according to my training data. Okay? So the expression over the right side of this slide are MATLAB code. I just want to see, okay, we can put 3 numbers, 3 data points all together as a matrix. And this matrix can multiplicate with the weight matrix of 2 sites. And each site will generate different kinds of output using the sum function. So the active function I use the sum function in this small example. Sum function, if you have a negative values, give you -1. Positive values give you positive. 0 maybe give you 0. So this sum function, just for illustration purpose, okay? So you change the weight, you change the numbers here, allow you to give the correct answer for the training data set. Then you fix these weights. This weight is the number you fixed, and your model is trained. You are done. You can deliver this weight and the structure gave to your customer. Say, okay, here is your classifier, and you're ready to use this machine to run this machine on your new data, and it will give you the output. If the model is good, the parameter is good, the training data is high quality, you should be able to make a good classifier. Okay? Any questions for this? So that's why people are so excited when they know there's a better way to do the matrix multiplication to save the computational time, because each step in the neural network is a matrix multiplication. This is so it's everywhere, and you can imagine how large the scale is if you use hundreds, thousands, millions of neurons in a deep neural network. And there are so many data points to training. And also, I will talk about the structure once you have multiple. So this is just one neuron. When you have a lot and make a network, each neuron have one output. The output will be as an input for next level of the neuron. So you can think about if you have another 4 neurons here, you can train this input, give to another set of the weight to second neuron. You can give another set to this neuron, give to again. And you use this 4 output. It's 4 outputs into another level of neuron. As an input, you can give to do the same thing, aggregate information again and again until you reach to the output. Then you can summarize. At the last step, you can aggregate information to give the output. Output will be the labels, okay, classifiers. So this makes a neural network. When we use ChatGPT language models, it's based on this framework, but it just uses different framework infrastructure. The topology inside is, they have the transformer topology. There are some attention. Attention is a way to label or set up the parameters. So there's some terminology in there, but the whole idea is the same. You start from the building a single perceptron and then start to propagate and add up layers, expand the scales, and give the better training data a large amount, and refine the output. So the output can be category, and if you think 1, 2, 3, 5 different category, when you have enough category, you can build a regression, because when you refine enough, those categories become continuous variables. So this slide discusses one of the activation function, and it's called sigmoid functions. So this function is linked with the logit function. Later we will also mention their inverse relation functions. So the logistic regression also has been used in this activation function to in the different scenario, maybe give you the better results. There you can have a linear classifier, and the activation function is linear and also nonlinear. So in this case, you have a hard decisions. Once you dot product value is greater than certain number, and then you switch to this category, and then otherwise is this category. Or you can have a continuous increase of being to be another group. Or when you have a logistic regression, and this is make a soft decision, there's the likelihood to be 10% to be 90% to be that, 10% to be this. This will be useful for you if you have multiple layers, then you translate, transfer information from this logistic regression, the probability, to next level, and then to make the final decision by aggregating multiple lines of evidence. So this is still the same thing I'm doing here, and just write out. You have 3 numbers and 3 weights, and then you do the dot product, give you the output of y. Okay. You train the model, and then. So in this case, we are going to see how the model, how this value should be updated. So I'm writing as an example in here. I just write 2 examples: data set 1, weight set 2. But in real application, you don't want to manually change it. You also don't want to randomly update until you get the best result, because that will take long to train. Okay, you don't rely on the chance to output the best. You need some feedback. You train the data and get output and see how you update those values. This deals with called perception learning. The source code looks like this is a Python code, and this is a MATLAB code here. You see, we start to do a loop. This loop will change the vector of your weight vector. It will update. It updated by using this, okay? There's and then after each round of this update, we will compare your previous step values of those weight with the current updated ones. If the difference is too small, when you compute the norm of this difference, then we stop. Then how you update this in the middle, how they update it, there are different ways. Okay, so this is a very simple way you add this small perturbation. To update your weight until your weight from a previous step and this step not change anymore. So they converge at the end. So this illustrates from a perception to the neural network. So if you consider this as the input data, then you give the input information to multiple neural perceptrons, okay, and make them the first layer of the intake of the inputs. And also you have multiple sets of this weight, and then each neuron will aggregate information from the input data differently and then move to next level. To do this, you have multiple neurons and multiple weights, so different sets, and your input. So this operation is vector multiplied by the matrix. Okay. In my case, I only have one perceptron, only one neuron. So I use this dot product between 2 vectors. But if you have 4 neurons, okay, you have multiple vectors to give you this matrix of weights. Then you just do the vector which input data multiplied by a matrix which multi-neurons and the weight matrix to give you the aggregate information to prepare you as an output for the next level. Does that make sense? And you can change the number of neurons. In this case, you have 4. If you make 5, you can still make it, okay? Because you just train. So the 3. So there's a variable is a 3 variable. So input. One data has 3 variable. Then you can put another layers, another neurons. So you can make from a 3-dimension input, use 5 neurons, each aggregate information differently, and pass on to next level, another 5. You can make these layers of neuron numbers, different layers of neural numbers, flexible. You can expand, you can contrast. So you can see we can have input, maybe a high dimensional data, but we use less neurons to learn. Okay, in that case, your input is very long vector. Say in the single cell data, one cell have 5,000 genes to define cell property, but we gave the 5,000 inputs into the neuron, and we compress them into 1,000. You use 1,000 neurons to aggregate information, and you basically reduce the dimension, and then you can further reduce to 1,000 to 500 neurons, and further reduce until maybe 50, and then when you really want, you can expand from 50 to 100 again, to make another layer to make 500. So it's very flexible. So that's why you start with high dimensional, you can reduce the dimension, you can expand, you can have a neural network, deep neural network, and then you have different layers. So how we construct this is kind of art. It has, if an experienced engineer, software engineer, when they do this job long enough, they know, okay, I want to use these numbers of the layers, and then each layer we have how many input, how many output. So to construct the neural network. So the one I just described is called, so this high dimensional data compressed into low dimensional in the middle, so there's hidden layers of neural network. And then you do expansion. So for example, 5,000 genes, one cell, and it becomes 500, and then in the middle, then you expand it to 5,000. So this framework is called encoder-decoder, so auto-encoder-decoder. What you want to do in this case, you want to compare the input with output to make them as same as possible. So for example, I have 1,000 cells, and each cells have 5,000 genes to define. So I have a matrix, single cell data matrix input. So if I build a neural network, so this neural network is from the bottom to top, but we can flip it, and from left to right the same. So let's give 5,000 genes to the neural network as the input. A neural network this autoencoder will learn the features of those and compress it into, say, 500 neurons, and then 150. So you make this information condensed as much as possible. Then you expand to 100, 500, until 5,000. So you build a neural network, change the parameters of those weights of each neurons until once you have an input looks like this, your output very similar to it. So your training is a process of the compressed information and then expand. So it's just like encoded information and decode them. Why you want to do that? Because we want to use the information in this low dimensional intermediate steps, those neurons in these hidden layers, to capture the features of your input. And this can be reduced to, say, 128 neurons Those weight is what we need. We can build the classifiers using those. So this idea is used in most recently publications. So this last month, it's a Nature paper. They introduced a deep neural network framework. It's called a SC similarity. It is a foundational model for scaled single-cell search. It's classifiers to give you. It's trained with millions of labeled cell type. It says 23 million cells. So they basically visit the geodatabase. You know how to visit the geodatabase and get the high quality single cell data and manually label them, okay, different cell type from humans. And then they build a human cell atlas. So there's a large collection of different cell types and normalized and QC and high quality inputs. Then they train this encoder-decoder framework. You gave the different cell type and let the neural network to learn, okay, to label it. So that's figure 1B of the paper, okay. Collect data from human different tissues and do a good job for the annotation, different cell types from different tissues, and also use standardized terminology to label the cells. Okay, you don't want to very weird name, but you standardize the official name for the cells. Then they start to build something like the training data. This training data include three cells for each training small data. 3 cells, one is called anchor cells, which is a place where they will use as anchor, as a reference later. And then use a cell which is a positive, is belong to this category, same category as your reference, the anchor cell. And then use a negative cell which not belong to the cell category of the anchor cell. So this uses this triplet. This is 3 cells, just like what we input here. The example I draw that in the blackboard. We have a -1, +1, or +1, +1, -1, something like that. Same idea. So they put this in, okay, the three sets train at the same time, not every time one cell. So 3 cells together give the input layers and learn and output the label. If it's correct, then the weight will keep that. If not correct, weight will change in the middle. There are many W's, okay? There's so many weight values, so we can tune them until you reach that level of the accuracy. Then the algorithm stopped training, and you can use this as a direct train. But also, what they want is the vector, those numbers, this weight. For each cell input, they will aggregate information and make a vector of weight in the middle, 128. Basically, compress the gene expression profile for every single cell, no matter how many genes in there. They have a set of the genes, and input as a output will be a vector of 128. So any single cell put in will be converted into a vector with 128 numbers. Okay, so now you can consider this whole framework after training as an encoder. So now the encoder is built. So I use this as encoder. So this is a set of genes you put. So this is cell X. Then you have sorted gene, gene 1, gene 2, gene 3, gene N. So this list is standardized, okay? If you have input data not in this order, you just sort your cells, your genes in this order as the input. Some cell type may have a missing value, or this 0, or maybe not, but at least you have a vector of data. This vector of data will be compressed into a vector of 128. So this is vectorized. So 1 by 128. You give another cell, this is cell X, cell Y, you get another vector. So it's become a information encoder machine. And once you have that, you have embedded 128 dimension of space, and then you can start to compute the distance between cells after you compress them. Okay? So this encoder, this compressor, contains all the W's, all the weight. You have been trained with all the human representative cells. So this encoder is not randomly encode your information into 128 dimension space. It uses information learned from all the tissues, all the different cell types. It will give you, if you give CD8, CD4 T cells, these two vectors will look highly similar. If you give the fibroblasts and the T cells, these two vectors will be very different. So you can use the output of this 128 vector for each cell to start to calculate the similarity between cells. Okay, similar cells will be close to each other. Distance is small. So we know how to compute the distance, okay? They use cosine distance. So because you have anchor cells, so these are reference. So use that reference as your label. Close to this reference cells will be labeled at the same type. Okay, so that's the whole framework. So this is a major features of this algorithm. So embedded into a low dimensional space. That's what I described. Many genes, profiles for each cell being compressed into the 128 space. And then the training would use the triplets loss function. So when the training, they provide reference, real, same category, different category. Use the cosine distance as a similarity score, okay, to compute. If you have cells, cosine distance is small in this 128 radial dimensional space. These two cells may be the same type, depends on the distance to the reference. So the distance used here is called metric learning, because when you see the metric learning, it's a machine learning field. Use distance to decide the outcome. So we can try to download the model on our computer, but the model is big enough. The model, when we say download, it contains this W. It's a set of weight for the weight has been trained. When I say we are going to use this model, that is, we prepare the input cell with a profile, give to the model. It will compute, give it up this, and then we will compute this, see which reference is close to our new provided cells, give you a label. But this model, the size required 32 megabytes RAM. So most of this computer have 16 gig RAM, so we cannot run this on our local machine because the parameters is too many parameters to load into my computer. Okay, so as I said, we need to have a, when we prepare the input, in this case, single cell is easy because each cell is defined by a vector. But if you have a picture like dog and cat, how can you transform this into a vector and to give to the machine? So convolutional neural network will take this picture and to find a way as a scan, to sliding window, to scan the picture, to compress information to make it ready for the neural network to take as a vectorized information. So the convolution, the process is this sliding window, basically scan your high dimensional picture information to convert that. So we will watch this process just before the break, and then we can so this is how the convolutional neural network will work for the first step, to scan your picture. In this case, there are 9 pixel small windows to scan and average or sum the values and put into another layer, and then scan again, and then compress the information to pass on into the neural network. So a lot of local information will be learned, features from the picture will be learned and transformed into the next level of the aggregation. Okay. Okay, so this is just a principle of the convolutional neural network. We can somehow, for example, if you have an input, you have a matrix of cells, and then with the genes, you can consider this matrix as a picture. Okay, you can do this convolutional neural network to scan the picture. Give you some idea. So there are some different ways to deal with boundary effects. Okay, you have so some details in the convolutional neural network, the process of data. Okay, so You also heard about the deep learning. When we talk about deep learning, basically we're talking about the neural network-based machine learning to do the classification, but they use more layers of neural network. There's nothing special. Okay, it's time to the break. Let's see the time. 5 minutes, okay? Okay, let's move on. So the neural network is the most important concept. If you don't know before, I hope you understand the principle right now. The rest of them are just a terminology introduction there. For example, the k nearest neighbor algorithm. So this basically you start with the training data already. So here you have purple and the yellow data projected in this search space. Then you have your new data comes in and as a location located in the middle of here. Then you have to decide how big of the neighbor you want to look at. So when you set a k equals to 3, then this will find the 3 nearest neighbors. And because in this neighborhood contains 3 neighbors, there are 2 purple ones and 1 yellow. So this new data points belong to purple group. When you decide to use k equals 6, in this case we will consider 6 nearest neighbor. In this radius, this community, 4 belongs to yellow, 2 belong to purple, then your new data points belongs to yellow by vote. So this is a very simple algorithm. There's no parameter. They just define the distance between the test data points with the training data. As the training data has been labeled, then it's easy to do. So this is a lazy learning algorithm, store all the training data and in the high dimension also. You need to define the distance. So logistic regression based on logit function, which gave you in this form, the logged form of the odds ratio. Basically, you have a p which belongs to this category, y minus p belongs to that category. So this function, then you model this using this combination of these parameters. Then you have this soft cut, and when you have the value in between, in this case you have 50-50 belong to each different category. Once you moved your measurement, then you either more likely to be the category on the top or other way around. So this logistic regression is one of the methods people use a lot once you have this particular problem with this 2 classes data, just one feature. So very quickly go through the next one. It's called the linear discriminant analysis, LDA. So LDA is very much like the PCA, okay? We want to find an axis. In this 2D space, we want to find one major component, one new axis. Basically, we want to rotate the points. So in this time, we are going to not only, in PCA, you know, you want to rotate your data so it can display the most dispersed projection onto your main PC. But in our case, in this case, we want to rotate all the data points. We want to projection to be more separatable between 2 clusters, okay? So when you consider 3D, in the 3D, we want to find a first 2D to define a plane. This plane once you project your data, they are most separable in this 2D space. So how can we make that? If you consider this blue and red points as a unit, as a category, as one category. In that case, you want to find the dispersion, which your first PC could be, let me see, could be here. So if you don't consider two clusters, then your PCA may give you this as a first PC, because you project on them, the cells or data points are more separated. However, what we want is something like this. We want to, let's see, I don't know which one is. So we want them to be between group, the separation between group is the largest. This is not a good example. I don't know which one is the LDA will found. But the idea is you rotate your data. Not only consider separation between data point itself, but between the categories, cross categories, cross groups dispersion. Okay? This is a better example. This, what I'm talking about is this. If you run PCA, you will get a PC, first PC, x-axis, because you separate data points. But LDA will find this as your first PC, because once you project your data onto them, the green and the red two clusters of data points, their projection are most separated. Okay? If you do this, consider no difference between two categories, then your projection gave you the most separated points overall, but you two clusters, data points belongs to the cluster overlapped, so you cannot give the best separation as clean as this PC will give to that. So you can see LDA is very similar to PCA, but when we do the rotation matrix, we want to make a projection most separatable for two categories of data points on the projected space, rather than to give you the overall largest dispersion as a PCA does. So if you look at the algorithm itself, it's very similar to PCA. So you have this centered your clusters, but then you want to compute within the and between cluster scatter. And also, once you have these two similar as a covariance matrix, you do this decomposition of eigen decomposition, and then you get your PCs. So this is a covariance within the within the group, and that's a between group. So you compute the variation of data inside the group, and also cross between the groups before you do this decomposition. Then you get the vector from the eigenvectors, which found its largest eigenvalue, corresponding vectors. Okay. This is an example in the source code in MATLAB. PCA, you only need X, but in LDA, you need to give labels for your data. Labels is, okay, so it can be multiple, okay? In our example, we only use red or green, but if you have another category, you can build that in, as long as each data point is labeled with one unique label. Then you compute how many clusters you have, and then you start to define the within the cluster scatter and within and between classes. So here you loop through all the variables, I believe. So at the end, you compute these two matrices, then to do the decomposition. So this is called generalized decomposition or eigendecomposition. You take these two matrices and then get this lambda, which is eigenvalues, and sorted to get eigenvectors, which are larger ones. You use this as a rotation matrix to rotate your original input. The output will be easy, is projected, will be easy to separate between different groups. Example, PCA try to find the projection, try to separate this data, and then LDA try to separate between groups, but make this within group separation small, but between group separation large. So we will play with this example in the computer session Thursday. Okay, so you also will have the Bayesian framework when you do this classification. So this is based on the Bayes' theorem. And basically, you want to compute a posterior probability and using the likelihood and also the prior probability. This example is a small illustrative example to see, okay, I want to find the new data like this. It belongs to yellow or green. The first I will compute, see, in total we have 17 data point training, and the 7 belong to yellow group, while 10 belong to green group. So without knowing anything, this is my prior knowledge about the majority of the data points belong to green. So we have a prior probability which is larger as a green, which is smaller for yellow, without knowing anything. Okay, this is, then we compute the evidence. If I look at this neighborhood, then so that's my prior knowledge. Then the likelihood. Then I make a calculation and compute the posterior probability is green. It's like this, and the probability being yellow is like this. So this is we use prior probability and the likelihood. This is another example to use naive Bayesian classification. You consider this training data documentation 1, 2, 3, and to 6 to see whether this is spammed or not spammed email, okay, message. So that's your training data. Then you look at the probability of being a spammed email and how many of them have this keyword or this phrase. And then use, this is like a probability, prior knowledge. And this is the likelihood. So if you consider the likelihood of being spam with this term, so through this, using this framework, we can compute two posterior probability when you have this phrase, whether the probability to be a spam or not, the posterior probability. This is an example. It also uses the frequency of words table. Okay, so if you have a null, for example, it indicates some urgent and it's being spammed. The frequency is higher than being in the non-spam. So those kind of information we take into account. I will show you an example at the very end. We use the frequency table to see, okay, this is a marker genes, for example. It expressed in one cell type, not in another, or some of these marker genes indicate multiple different cell types. So the frequency table, we need to take that into account as well. There are many other that you can see the reference, and you can take a look. So how can we have a reasonable evaluation for how good or how bad a classifier is? So there's several methods, and then this is called cross-validation. Cross-validation, give you an example, is to separate your training data into, say, for example, you want to do the tenfold CV. CV stands for cross-validation. In that case, you will randomly assign, you separate, you do 10 rounds of tests of your classifiers. Your first round, you will randomly select 10% as your test. So this will be test. And use the rest of 90% of your data points as a training. And then after you train your model, use 90% of the data, then you test with your 10% of the data. And then you get your accuracy. See, okay, in this case, I can predict 90% cases in my test. And then you do that again, randomly assign 90% as a training, 10% as a test. See, I think I made a mistake. So you don't have to be 10%. So it doesn't has to be 10%. You can see 20% as a yellow, as a test. It's okay. 20%, 80%. As long as you make a reasonable decision, say I want to use a part of the as a training and a part of the test. Depends on how many training data you have. Make a reasonable set. When you see tenfold, has nothing to do with percentage of here, but it's how many rounds of your random test you want to do. So you do this 10 times, then you take average of your validation accuracy as your final. So average this 10 round as your final accuracy. So I see you can see, okay, my model has been tested with the final accuracy, say 93% after tenfold cross validation. Okay, people in the field will understand what you are doing. You can use a 5-fold CV if each time take too long, sometimes not reasonable to run 10 times. You can see 5-fold cross validation. The accuracy my model is 91%. But people may ask you how you separate your training each round. You can see 80 training, 20 test, 20%, 80%. Or some people use 30, 70. Okay. So these are the idea of the cross validation. A robust classifiers should have a very good accuracy in average accuracy in this cross validation. You cannot see, okay, I trained my data using this setup, so 20% of yellow and 80% of blue, and in this case I get 99% accuracy, and I just report this. In this case, there's no cross validation. And you only report the highest accuracy you can get, but people only trust you. But you have to do this multiple fold of the cross validation. You take an average, and this gives you some idea of how robust your classifier is, not based on the training data set to be stratified just by chance and give you the best results. When we see accuracy, there's or recall or sensitivity, there are definitions of what is accuracy. So you have to deal with this 2x2 cross tables. This positive, true positive, true negative. So you want to have a bigger numbers in these two locations, where you want to less false positive, false negative. When you report accuracy, will be the summation of a true positive, true negative divided by the total. So there's a percentage. You predict something, either yes or no correctly. Well, the false positive rate, the recall, is the ratio between true positive divided by true positive and false negative. So true positive, false negative, basically you are doing in these two numbers, the ratio. Sensitivity, specificity, they all have their definition from this. So those are the numbers you want to consider when you report how good, how bad, or using those numbers to give people the impression. Another way is to call ROC curve. I don't know how many have you seen this before. In this ROC curve, you have the space defined by false positive rate. False positive rate is also defined by the 1 minus specificity. Okay? If you look at the definition, it's the ratio between these two numbers. A good model should have a small false positive rate. Okay, small positive rate means small positive, small false positive. So something was not, so it's not there. You don't want to predict that. You want to have a low report to be yes to that particular case. So you want to small this number. Well, in your y-axis is a true positive rate. So you want to as large as possible. So if you have a model when you trained in the case of your test, sometimes give you this value, sometimes give you this value. So at the end, you run multiple tests or different setup. At the end, you get a curve like this. So this curve should be pushed towards left-right position. And then you calculate the area under the curve. The larger, the better. You may have a model which, when you train the data differently or set parameters slightly differently, you get a curve which close to this diagonal line, then the underlying curve is small. If you purely random predictor give you the position which straight line here, okay? So you don't want to your model to be close, the curve close to this diagonal line here, because under the curve area is too small. So you want to report this under the curve area as large as possible. Your model is sensitive enough, which close to this side, and also specific enough in order to give you this large area under the curve. All right. So this is how to read ROC curve. I will mention several single-cell classification tools which has been published over the years. The latest update, what I gave you, is a SC similarity based on the neural network, which is this. Okay. So this is good enough to do the classification. The rest of them not in the mainstream right now. They're correlation based, SC maps, cosine similarity based, then SC prediction using SVM, support vector machine, Surret, and use random forest. This is a list of the table from a review paper to evaluate single-cell classifiers for the single-cell data set. So those are the packages, the methods have been tested in this review paper. Look at the algorithm. There are k-means, the KNN, cosine distance. There are SVM, there are Spearman correlation based. This is a random forest based. Yeah. Okay, and they have a different inputs. Mostly in language of Python or R. So this is neural network based. It's based on the cell similarity. So all this require the training data. So you have to have a reference cell type. This is a pipeline in the SC predict in the supervised method. So the training have labeled the cells with the gene matrix, and to do the PCA first. Do the feature selection. I believe their feature selection based on how well the separation of different cell type. So this will be very similar to LDA, this step. And then this k-fold cross-validation. So you should start to understand what they are doing here to separate the training data into trained and tested, and found the best model, which is defined in this training model here. Parameters, performance. Then once you have your input data, we start to prepare the matrix and to do the rotation. And then to use this function, which has already been trained, to use this rotated feature extracted matrix, which the gene expression has been replaced with the PC. And this, so this is similar to the similarity, right? The same idea, you project after training. Yeah. But I haven't seen the real application for this. So this is a different package. They also consider the across the different species. So cluster, cell clusters, and build a class. They use random forest model. Yeah, so basically this is the different pipelines people use to do this. This is another paper and also using random forest and emphasize pathway based. It can do clustering, classification, and build interaction network. Okay. This is a paper I make as example study published in Nature Medicine. This is an example of using the manual annotation. I think they not rely on any trained model to do the prediction. I think this the biologists know their systems, and they use the gene expression programs, cell cycling, immune regulations. So they know those key marker genes. So manually label their cells. So resident memory T cells. Remember last computer session, I played this Michael manually annotate his cell type in the clonal sites. He used a lot of these marker genes to look at each clusters, subclusters. So the similar procedure has been used in this paper. So I want to bring that up. In real application, you may not rely on any of these trained models or methods. You can just use marker genes, check clusters one by one, assign them, label the cells using your knowledge. Okay. So at the end of today's lecture, I want to mention PangalawDB. So this is a database of marker genes. Marker gene database. It also programs that can use these marker genes to label the cell, give a cell type. But this is a Swedish group. The research group ran out of the funding, so their PangalawDB database has stopped running. I don't know whether the PangalawDB website is still available there. It's so-called secondary database. It's pretty good. They process the geo database and also the database of single cell data from the European government-based these data sets already. They synthesize this process in the uniform way, so you can cross-validate or compare the cell type annotation even across different studies. The marker gene database, they have a 180 something different cell types. For each cell type, there are several marker genes. If your cells have a high expression of those marker genes, you should label the cell type in the cell type given. So it's very good data set and also database, but unfortunately has been discontinued. The publications is there and the website is there. So I want to mention the algorithm of PangalawDB use the marker genes expression to assign cell type, because this is the algorithm I re-implemented in the MATLAB, and I also use the PangalawDB database to assign the cell type. So in the interface of MATLAB, when you click automatically assign cell type, I use the algorithm here. So this algorithm consider first to give each marker genes, so this is genes, a function, use this function to give a weight of this gene. So if you look at the marker gene database, there are maybe several hundred of the unique genes, but some genes, so maybe, for example, ATP3, it may appear in several different cell types. So the specificity of this gene not high. So we want to weight this gene level a little bit lower. So this gives you the frequency, basically, of the gene appeared in the whole database. If a gene very specific is like a CD8A, it's only appeared in CD8 T cells. It's very unique. The weight of that gene will be higher than compared to another genes which appeared in multiple different cell types. The relative weight for that universal gene, marker genes, will be lower. So that's the first step, to give the weight for each gene in the database, a different weight depends on their specificity to be as marker genes uniquely assigned to the individual cell type. So after that, we will start to look at each different clusters differently. Say cell cluster K, so that's a K. And cell cluster K belongs to cell type J. Okay, so for each clusters of cells, I will assign values for this cluster to be cell type J, cell type 1, 2, 3, until J. So if I have 128 different cell types in my database, I will assign 128 different numbers for a cluster of cells. The score to be the highest is most likely to be that cell type. So I'm not computing just one cluster, one cell type, but I compute a score for all the cell types in my database. So how can I compute this score for cluster of cells belongs to one of the cell type? I use the average of marker genes in the cell in the cluster. So that's the value of Z. Okay? So Z means the average expression of all the cells in cluster K, which are the marker genes, N marker genes expression in the cell type J. So I take the average and then average and also weighted by this particular marker's weight. When I say average, it's divided by the N. Use the square root of N to do the average. At the end, I will build a table, a cell type table. So cell type 1, 2, 3. I don't know how many, but it's a J table. Cluster 1, 2, 3, K. In my data set, different cluster. Each cluster I assign to the different cell types. So this is the DB scan, DB, Pangalal DB, cell types. So for each of this cell type, I gave you a Z, the score. So this score is computed by different N different, so you have N different markers. 1, so marker genes. Marker genes expression, summation. So basically you add them up to give you Z. So if a cluster have a cell type N, cell type 1, marker genes expressed high, so you have a higher value here. And for cell type 2, the marker genes expressed less in the cluster, so this value will be low. So this will be different, maybe some low, some high. At the end, you found the highest value in all the cell types, Z value, and gave this cluster and this cell type. And the cluster 2 may have this cell type. highest. Cluster 3, maybe this cell type is the highest. At the end, you can give the top highest Z values to different clusters. Oh, that's all for this week. I just gave a quick review. You have a neural network framework and different other types of the classification method. And you use cross-validation and ROC curve, sensitivity, specificity to define the metric to evaluate your classification method. And then in real application, we have a database-driven and compute the frequency and the summation of the expression marker genes. And also, I encourage you to use manual annotation in the real application. Okay, and that's all for this Tuesday, and see you on Thursday computer session.